seeks to understand how species respond to major geologic, climatic, and anthropogenic events, and how different species traits mediate those responses. To do this, I use methods from phylogenetics. So perhaps the first thing you think of when you think of phylogenetics is inferring really big trees for lots of taxa, and certainly that's a part of it. But we can also use those trees to ask questions about how different things have impacted diversification. So for instance, we can look at diversification rates through time and try and associate those with environmental factors like surface temperatures. Of course, this is extremely difficult, but we still can learn a lot by doing things like this. We might also want to focus in at a narrower scale. So for example, this is a tree of mammals, so I'm highlighting bears here. And this is an example of a study where they focused in, instead of looking at a tree of mammals, they focused in on bears, and they tried to understand the history of these bears. So they tried to understand the importance of integration between polar bears and grizzly bears, and also how effective population sizes had changed through time. And so by looking at things at different scales, we can answer a lot of different questions about how different things impact species. And in my lab, we use a combination of field work, lab work, and analytical methods to ask these questions. But today I'm going to be focusing mostly on analytical methods because this is what I've been thinking about a lot in the last couple of years. And so I think one of the most important things here is that biology is extremely messy, and trying to make inferences about things that happened millions of years ago is incredibly difficult. And so a lot of the work in my lab tries to understand when the methods we use can be misleading and how we can improve them, and also how we can most effectively use the data that we have to make accurate evolutionary inferences. And so I'm going to talk about these two things today by focusing on three different small research stories. First, I'm going to talk about the effects of selection on inferences of migration. Then I'll talk about some machine learning approaches that can help us do a better job. And then finally, I'm going to shift gears a little bit and talk about the data we use instead of the models we use, and how we can use paralogs in phylogenetics. And so we'll start by talking about selection and migration. Lots of evolutionary biologists want to know whether gene flow has happened between closely related populations. We want to know this for many reasons. So for example, people who are interested in speciation want to know whether species formed in the presence or absence of gene flow between populations. And we also might want to know this so we can develop appropriate null models to do things like detect loci under selection. So unsurprisingly, a lot of people want to do this, so there are lots of methods for doing this. And these methods include things like methods based on site frequency spectrum, like FastSim-Col2 and DOTI. If you're not familiar with the site frequency spectrum, it's a summary of biallelic data. So if we take SNP data, we can count. So let's say we have a SNP and there are two alleles. We can count how many times the minor allele occurs in each population and use that to summarize the data. So for example, in this site frequency spectrum, the bin here, I'm not actually sure if you can see my cursor. I think you can. So this bin corresponds to SNPs that occur where the alternate allele occurs 0 times in population 1 and once in population 2. And so this is just a convenient way of summarizing data. And there are a lot of very fast methods that can use these data to infer things like migration rates. There are also methods that use our sequence data directly and more effectively use all of our data. And these include methods like BPP and its implementation of the multi-species coalescent with integration. And I'll talk a little bit more about these methods as we go, but the basics here are that these methods allow us to ask whether or not migration happened between closely related populations. But there's a huge caveat to these methods, and that is that they assume a neutral model. And by that, I mean that these methods assume that selection is not shaping genetic variation in the genes that are being analyzed. But we know this isn't a very realistic assumption because it's become increasingly clear that selection shapes large parts of the genome. Even regions that are not directly under selection are often linked to regions of the genome under selection, and this can have important impacts on the genetic diversity at these loci. Not only is ignoring selection unrealistic because selection impacts large parts of most species' genomes, but we also know that it can mislead demographic inference. And so linked selection misleads inferences of population sizes. There have been several studies showing this. And we also think that selection may mislead methods for detecting migration. But we know less about how selection misleads the most popular methods for detecting migration today, particularly when we're thinking about realistic models of selection, because there haven't been many studies of realistically modeled selection and its impacts on these methods. And so that's what we set out to do. We wanted to ask, are these popular methods misled by biologically realistic selection? So to do this, we simulated data under several different models. We started with a simple neutral model where we had constant recombination and mutation rates and no selection. And so this is the model that most of these methods assume. We also had a model with background selection where we still had constant recombination and mutation rates, and 75% of mutations were deleterious. We had a complex neutral model with variation in recombination and mutation rates across the genome, but no selection, and a complex background selection model. And here we have variation in recombination and mutation rates, and we have selection. This time, though, the selection only happens in exons and conserved non-coding elements, and selection is stronger in exons. And the genomic architecture here was based off the Drosophila genome. And then finally, we had a complex sweep model. This is built on top of that complex background selection model, but we also have a beneficial mutation that's introduced and allowed to sweep to fixation. Okay, so we simulated these data. We used forward time simulations in SLIM. If any of you have done forward time simulations, you know that these can be rather slow. So we used an approach in PySlim and MS Prime that allowed us to overlay neutral mutations. You don't need to know much about what that means, but it just makes these simulations a lot faster and more feasible. We looked at 3 different divergence times. I'm going to focus on the deepest divergence time. And we set recombination and mutation rates as shown here. Okay, so we used 3 methods. Remember, FastMCoL2 and DOTI are methods that are based on the site frequency spectrum. And using those approaches, we can do model selection. So we can compare models with and without migration, and we can estimate parameters. And then we also used BPP. This is an implementation of the IMSCI model, and it's a Bayesian implementation. So we can estimate parameters, and we can look at our posterior distributions of parameters and ask how often those posteriors for integration probabilities include 0. Okay, so I'm going to go through each method in turn and tell you about a little bit more about how it works and what our results showed. So in FastMCoL2, we find parameters that maximize the likelihood of our site frequency spectra under our specified models. And in this case, our specified models include a divergence-only model and a model with continuous gene flow. We compare these models using likelihood ratio tests, and we also looked at our parameter estimates under each model. Okay, so these are the results of the likelihood ratio test. On the y-axis, I'm showing the percent of our replicates. Here are the results for the simple genomic architecture, so constant recombination and mutation rates. And here are the results under the complex genomic architecture. Light blue means that we failed to reject the divergence-only model, so we don't support migration. And it's important to note that that is the correct answer here. There is no migration in these simulations. So when we have a simple genomic architecture, even when there is selection, we never reject that divergence-only model, so we're doing a great job. But when we overlay that onto a complex genomic architecture, then add selection, we always reject that model in favor of a migration model. And so we're getting things wrong 100% of the time. We can also look at estimated migration rates, and we see the same pattern. When we combine a complex genomic architecture with selection, whether that's background selection or selective sweeps, we infer non-zero migration rates. However, those rates are quite low in absolute terms. So in light blue, I'm showing rates that were estimated for data sets that did include migration. And in pink, I'm showing our estimates from our data sets without migration. And so even though we're inferring these to be non-zero, they are low in absolute terms, which is slightly comforting. Next, we use DOTI. DOTI is really, really similar to FastMCoL2. We're still calculating the likelihood. We're finding parameters that maximize the likelihood of our site frequency spectrum. We're just using a different approach to do that. And we're able to compare these same 2 models and estimate parameters. Okay, so again, here are the results from the likelihood ratio test. Light blue is good. We didn't reject the divergence model. And we do a great job under the simple model. But when we combine the complex model and selection, we always reject that model in favor of a model with migration. Again, very concerning since there is no migration in these data sets. And again, if we look at estimates of migration rates, we see the same pattern. Rates are elevated under this complex genomic architecture with selection. But yet again, we have this caveat where if we actually look at those migration rate estimates, they're rather low, and they're much lower than rates we see in blue here for models that include migration. Of course, if we simulated data sets with less migration, we could make these results more similar, but still these results are quite low in absolute terms. Finally, we use BPP. BPP is a bit different than these other two approaches in terms of the model. The model has a single integration edge instead of continuous gene flow. And it's a Bayesian approach, so it takes a lot longer to run, but it's able to estimate posterior distributions of our integration probabilities, divergence times, and population sizes. And we look at those and record how often the 95% highest posterior density interval does not include 0. And so the idea here is that if there's no migration, those intervals should include 0. Okay, so these are the results for BPP. And so what I'm showing here, light blue means that those intervals did not include 0. So that's an incorrect inference. And light blue is a correct inference. Light blue means that our intervals included 0, and dark blue means that our intervals did not include 0. Interestingly here, we see that we actually don't do very well under the simple genomic architecture. This could be explained in part by the fact that BPP does allow for variation in theta across loci, which means that our complex model is actually a slightly better match to the model in BPP. And so the complex neutral model is actually the best match to the model implemented in BPP. And here's where we have the lowest false positive rate. When we add selection, we have increased false positive rates. Again, if we look at estimates of our integration probabilities, they're non-zero when we have a complex genomic architecture and selection. And in this case, they're also non-zero under the simple genomic architecture. But yet again, they're quite lower than estimates in the presence of migration. And so while non-zero, these inferences are low on an absolute scale. Okay, so we found that selection does lead to the rejection of these divergence-only models in favor of migration models in FastSim-Col2 and DADI, and it leads to 95% highest density intervals for integration probabilities that do not include 0 in BPP. So selection is causing us to falsely infer migration. It leads to elevated estimates of our migration rates in all of the programs, but even though these estimates are non-zero, they are relatively low. And so in the future, I think it's important for everyone to keep in mind that when you estimate migration rates between sister populations without considering selection, you should be really careful in terms of how you interpret those estimates. And I'm hopeful that the combination of machine learning approaches, which I'm going to talk a little bit about next, and more efficient forward-in-time simulators could allow us to develop approaches that work well in the presence of selection. Okay, so I just told you about how when our models aren't sufficient, we can arrive at really misleading inferences that affect how we think about the biology of our focal systems in important ways. Now I'm going to continue in a similar vein and talk about how we can use flexible approaches to try and accommodate these complex processes. Okay, so a lot of you probably are somewhat familiar with machine learning. Machine learning is a subfield of artificial intelligence that uses data to perform inference without any explicit mathematical models. When we use machine learning, we're identifying patterns that can be used to predict unknown outcomes. And there are two major classes, unsupervised and supervised, but I'm going to focus only on supervised machine learning for the inferences we're talking about today. In supervised machine learning, our goal is to learn to predict some output from some input. To do this, we need training data to learn the mapping between input and output. I can't learn things without having an example to learn from. And we're going to be tuning parameters to maximize our prediction accuracy. In population genetics, I don't know about you guys, but I do not have access to lots of data sets from slugs where I know the migration rates. I don't think any of us do. And so To get our training data, we actually use simulations under some model. This gives us training data with a known history. From that training data, we can extract features and use some algorithm to build a model, which can then be used to make predictions on unknown data, so empirical data sets. And I think a really important of this process is the methods that we use to get our training data. And so we're going to have to simulate these data sets under some model. But what happens when our simulations aren't very realistic? And so what if we simulate data under a neutral model, but selection is important? And this kind of misspecification, you can think of it as somewhat analogous to what I was talking about in the last section, where when our methods assume a particular model, it can mislead inference. When we train under a particular model, it could mislead inference. In machine learning, we think of these as domain shifts. So domain shifts are differences in the distributions of source or training data and target or empirical data. So our source data, that's just another word for training data sets. These are the data sets we would simulate in population genetics and use to train our algorithm. And our target data are going to be the actual data that we want to apply our algorithm to. So in our case, empirical data that you've collected from real populations. In machine learning, a famous example is digit classification. So there's a really large data set of these handwritten digits, and people build all kinds of networks that aim to decide whether this is a 2, a 3, or a 4, or a 9. And they train these algorithms, but then they might want to apply these to slightly different data sets that also have digits in them. And so let's say someone wants to classify this 2. This is actually from a Street View house numbers data set. So this is from a sign. And so in this case, our source data are handwritten digits, and our target data is from a sign. So these distributions differ. In population genetics, our source data perhaps was generated under a simple model with divergence between 2 populations. But let's imagine that in our actual empirical data set, there's gene flow from an unsampled population into one of those 2 populations. We call this ghost introgression, and it leads to a difference in the distribution between our source and target data sets. And so my goal here was to evaluate the impacts of a domain shifts on a convolutional neural network, which is just a type of machine learning algorithm for detecting migration. And so as a first step, I trained a CNN to test for migration. And so our CNN is comparing a model without migration and a model with migration. These are very similar to the models we talked about in the last section. And so these are our training models. And to evaluate the impacts of domain shifts, we developed 3 different testing models that differed from these models. And so the first one, we call it the misspecified prior model. And here we have more recent divergence times than we used in our training data set. Our second data set is a background selection model, which is very similar to the model we talked about before. And then finally, we have a ghost introgression model where there's gene flow from an unsampled population into one of our 2 sampled populations. Okay, so these are the results. Here I'm showing the results in the absence of a misspecification. So we just test this on data generated under the correct models, the models we use for training. When we generate data under a divergence model, we select the divergence model 97% of the time. And when we generate data under a gene flow model, we select the gene flow model 97% of the time. When we apply our classifier to data set where the prior was misspecified, so our divergence times were more recent, we find that when we simulated data under a divergence-only model, we always classified that as a gene flow data set, which is not surprising. We've made the divergence times more recent. This is what we would expect. When we generate data under background selection and run our CNN, we find that we have elevated error rates under both models. And when we generate data with ghost introgression, we find that data sets that were actually generated under divergence-only models are actually often classified as having gene flow. And so we are finding that, yes, indeed, these domain shifts are leading to inference errors. Okay, I've already told you that methods didn't work several times, so how am I going to fix it? So I'm going to tell you about some potential approaches that we're working on to try and design networks that are robust to these kinds of issues. Because in reality, when we collect biological data, it's never going to exactly match the model that we're using for training these networks. So we need to find ways to build networks that can work with this. The first option is to generate data under the correct model for training. And I'm not going to actually show you results from this approach, although we are trying it, and it does work. The important caveats here is that this can be really expensive. If we need to generate lots of data under selection, those simulations are pretty slow. And even though they've gotten faster, that's pretty intensive computationally. And more importantly, perhaps it's also impossible when we have empirical data, particularly in some groups where we know very little about the genomic architecture and the DFE, or the distribution of fitness effects and how selection acts on the genome. It's going to be really hard to design hyper-realistic models. And so we're going to have to work with what we have. The second option is to add noise to our training data. And so perhaps our data is just noisier than the training data, and this causes us to select more parameter-rich models, like models that include migration. We tried this by allowing for variation in the effective population size across loci. And so what I mean here, typically when I generate a training data set under a divergence model, I generate several hundred loci under that model. And the standard approach is to have a constant effective population size per replicate. So each locus is generated under the same effective population size because we think of this as a population level parameter. But several different people have tried allowing this parameter to vary across loci or genes, and this can approximate some of the effects of selection on the genome. And then finally, the third approach is to use something called domain adaptation. Domain adaptation aims to train a network that will work well in the presence of domain shifts, which is exactly what we'd like to have. And so here I'm just showing kind of a bare bones a layout of a network. So our network has some input, which is our labeled source data. So these are our simulated training data where we know which model they were generated under. And we have unlabeled target data. So these are empirical data, and we're trying to find out what model they were generated under. We have some feature extraction layers that we use to get features. And then we use these features and some additional layers of our network to classify. And this is what we call our classification task output. This is just gene flow or divergence in our case. To do domain adaptation, we're going to add additional layers. And so we could add what's called a distance-based domain invariant feature extraction layer, which sounds more complicated than it is. Essentially, all we do here is we're going to measure some distance between our training data and our empirical data, and we're going to try to minimize that data, minimize that distance. And so by doing this, we're trying to extract features that are invariant across our domains. And this should lead to a more robust classifier. We could also use what's called adversarial domain invariant feature extraction. Here, yet again, the goal is to extract features that don't differ between our source and target data sets. But we do this in a slightly different way. We have what's called a discriminator here. This is just another network that tries to predict whether an observation is from the source or training data or our empirical data. And we use what's called a gradient reversal layer. And all this is doing is saying we want to do poorly at this task. So we want to extract features that our discriminator cannot use to distinguish between our training data and our empirical data. And so both of these approaches try and extract features that are similar across our domains and should lead to more robust classifications. Okay, so again, we had these three options. I'm not showing you any results for the first option. I'm going to show you results from our noise classifier, where we added noise to our training data, and domain adaptation. Okay, so first here, I'm just showing you the results in the baseline case under our three misspecifications. Remember, when we have a misspecified prior, we do really poorly. We have 0% accuracy on data sets generated under divergence-only models. Our average accuracy is 50%, and we have really poor balance across our classes. With background selection, we have reduced accuracy compared to the baseline case, but fairly balanced accuracy between classes. And for our ghost introgression case, we have reduced accuracy, and we have lower accuracy on data sets generated under the divergence-only model. Okay, next we used our noise network, where we'd added noise to our training data. We find that we do slightly better under a misspecified prior, and we achieve slightly better balance between our two classes. Under background selection, our average accuracy is slightly increased, but we have we've really increased the discrepancy between the two models, so this isn't working very well. And under ghost introgression, we see a very slight increase in accuracy, and we see similar balance across our two classes, although we've flipped here. Finally, we tried CDAN, which is a domain invariant feature extraction approach. So this is our domain adaptation technique. We don't do well under the specified prior. We do worse than we did with our noise classifier. But under background selection, we see that our average accuracy has increased significantly, and we have good balance between our two classes. And so even though we're still not doing nearly as well as when the model was correctly specified, this is a pretty substantial increase that's promising, I think. And then in the ghost introgression case, we see the best performance. We see an increased correctness, so we're getting closer to those baseline values of nearly 100% accuracy. And we see really good balance between the two classes. And so we showed here that when the models used to generate training data fail to include important processes, it leads to domain shifts and inference errors. But injecting noise helps, but has limited effectiveness. And finally, using domain invariant feature extraction decreases our error rates under some specifications, but not all. In the future, I'm really excited to try and combine using more complex models with domain adaptation. So we could add noise and use domain adaptation. We could try to do slightly more realistic simulations and use domain adaptation, and that might increase our performance. We're also going to be exploring additional domain adaptation techniques and exploring alternate network architectures. Okay, and so I started out by telling you how our methods could be misled by things like selection. And then I talked about some flexible machine learning approaches and how these might be used to increase our confidence in inferences on messy empirical data sets. And finally, I'm going to shift gears. And the last thing I'm going to talk about is the types of data that we actually use. And so this is going to be a bit of a more positive story. So I've been talking about what happens when our models are misled. And now I'm going to talk about how we can expand the data we use and still make accurate inferences in phylogenetics. Okay, so one of the main promises of phylogenomics was that we would have more genetic data than ever before. And because of this, we would be able to resolve the tree of life. So on the y-axis, you see the number of genes in phylogenetic studies, and on the x-axis, we're showing the year. And so obviously, we have way more data than we ever did before, and it continues to increase. But what became abundantly clear as we collected these data was that gene histories often differed from the species history. And so imagine that we're sequencing data from human, chimp, gorilla, and orangutan. This is the accepted species history, but if we sequence any individual gene, we might get a quite different history. So we might get a history that matches the species history, but if we sequence another gene, we might find that gorilla and orangutan are sister species. And if we sequence a third gene, we might actually find that human and gorilla are sister species. And gene histories can differ from the species history for many reasons. It could be due to incomplete lineage sorting. So this is a process where polymorphism in an ancestral population leads to different species inheriting different versions of this gene that with histories that don't match the species history. So in this case, in our gene tree, B and C will be sister species, even though those aren't the species relationships. Introgression can lead to discordance. So in this case, introgression between B and C leads to a gene tree where B and C are the sister species. And gene duplication can also lead to discordance. So in this case, we have a duplication event in the ancestor of our three species. After this, we have a green copy and a pink copy, both of which undergo subsequent speciation events. And this leads to several different genes. So we have two copies in each species here. Some of these genes are paralogs, meaning they're related through duplication events. So the green copy here is paralogous to this pink copy because it's related through this duplication node. Similarly, the green copy is paralogous to the pink copy in species B. We also have orthologs, or genes related through speciation events. So the pink copy in A is orthologous to the pink copy in B because those are related through the speciation event. How do we deal with this gene tree heterogeneity when we want to infer a species tree? Typically, we accommodate incomplete lineage sorting by using the multi-species coalescent model, or a method that is robust to incomplete lineage sorting. We accommodate introgression either by testing for introgression after we've already inferred a tree, or by using models with introgression to build things like species networks. But when it comes to gene duplication and loss, the most common approach is to try and exclude any genes that have a history of duplication. In other words, we try to exclude paralogs. Okay, but there are a few problems with trying to exclude paralogs. One of the major problem is that it leads to massive amounts of data loss. You might think, I just told you we have more data than ever before, so who cares if we lose some of it? But even when we have large numbers of genes, throwing out anything that could be paralogous can leave us with very few genes. And so the most typical approach in phylogenetics is to use only single copy genes. And so we only use genes where there's a single copy in each sampled species, because it's thought that these are likely to represent orthologs, because if there's a history of duplication, we might expect to find more than one copy in some species. And so what I'm showing here is the number of genes on the y-axis and different groups on the x-axis. The gray bars represent the number of genes in which all species are present, and the black bars represent the number of genes in which all species have only one copy. So for example, for fish, we have a lot of genes sampled for all of our species, but only a very small number of those are present in only a single copy in each sampled species. And for some groups like plants, the situation is even worse. And so even when we have a massive amount of data, we might end up with not enough data when we throw away everything that's not single copy. Beyond that, it can actually be quite difficult to remove paralogs from our data sets. In this case, we have a duplication event shown in red with subsequent speciation events shown in blue. We can pull that out and show the tree here where we have copy 1 and copy 2. When we have a tree like this, it's fairly obvious that a duplication event happened because I have 2 copies in species A. So obviously we have some paralogs. But we also have gene loss. And so imagine we lost copy 1 from species A and C and copy 2 from species B. This is going to lead to a gene where we have 1 copy sampled from each species, but some of those copies are related through gene duplication events, not speciation events. And so it's actually quite difficult to make sure we're actually removing all of our paralogs. And so during some of my postdoc work, we set out to understand the costs and benefits of including paralogs in phylogenetic analysis using a simulation study and an empirical investigation. And so I'm going to talk about the simulation study first. In the simulation study, we simulated data using a program called SimPhi, and we simulated data with realistic levels of incomplete lineage sorting and gene duplication and loss levels ranging from 0 to 10 times those seen in empirical data sets. So we simulated gene families. Some of these gene families had no duplication events in them, and some had multiple copies per species. We then used several different sampling strategies to get data sets for downstream phylogenetic inference. First, we sampled only single copy genes. So these are the traditional genes used in phylogenetics. But then we sampled all genes. So regardless of how many copies were present in any given species, we just use the gene. We also had some other approaches, but I'm only going to focus on these 2 most extreme approaches for today. Okay, so here on the y-axis, I'm showing the Robinson-Folds distance from the species tree. So 0 means that we accurately inferred the species tree. This is the distance from the tree used in simulations. We used several different species tree inference methods. We had Astral, MPL, and NJST. So these are all methods that should accommodate incomplete lineage sorting well. And we also had Astral Pro, which should accommodate both ILS and gene duplication and loss, and FastMole RFS, which should work well in the presence of duplication and loss. In the case of the single copy genes, we find that the more genes we have, the better we do. It's not unexpected. This is still a fairly hard problem, but we do best with 250 genes. When we compare this to the results when we use all of our genes, we actually see extremely similar patterns. And so it appears that even when paralogs are included, we are accurately, and well, it's not decreasing the accuracy with which we infer the species tree relative to including only single copy genes. So our simulation study suggests that species tree inference is accurate in the presence of paralogs. But of course, simulated data sets are easier to work with and cleaner than most empirical data sets. And we wanted to investigate this in an empirical system. To do this, we used empirical data from 26 primate species and 3 non-primates, originally published in Vanderpool et al. This data set includes 18,400 genes that were sampled in at least 4 species prior to any filtering. But in the original paper, they only used 1,700 single copy genes that were sampled in at least 27 of the species. And so they lost a massive amount of data. We took this data set and we analyzed the single copy genes again as a baseline, but we also analyzed all the genes. So we included genes regardless of how many copies were present. And we used a branch cutting approach. So branch cutting approaches aim to extract orthologs from larger gene family trees. So for example, in this case, where we have 2 different subtrees, each with all 4 species present, we can cut out the left subtree and we can cut out the right subtree and use each as an ortholog for downstream inference. So by using either the subtree extraction, branch cutting approach, or all genes, we can include far more data. We're including almost all 18,000 genes using these approaches. We also used a couple of other approaches, which I won't talk about here, but the results were fairly similar. We used 6 species tree inference methods. So we used Astral and Astrid. These are both what we call summary methods that take gene trees as input, and they should be robust to incomplete lineage sorting. We used SVD quartets, which again is robust to incomplete lineage sorting, but takes alignments as input. We used concatenated maximum likelihood and maximum parsimony. And we used Astral Pro, a method that is designed to work in the presence of both incomplete lineage sorting and gene duplication and loss. So we had 6 species tree inference methods and 16 different filtering strategies. I only mentioned a few here. We had 2 different gene tree inference methods. So for approaches that relied on gene trees, we inferred those trees both using maximum likelihood and maximum parsimony. So we ended up with around 200 species trees. And I have some time, so I think we'll just walk through and compare all of those. Just kidding, I won't do that to you. I'll only show you these 2 trees. So I'm showing you the tree from single copy genes in Astral. So this is kind of our baseline approach, and just using all of the genes in Astral, which is one of the most, which is an extreme approach, right? I'm sure it's obvious to you just from glancing at these large trees on this slide that there are no differences. You'll have to trust me, but these trees are identical. And what we found was that all of our species trees were actually identical except for at a single node. And that node was involved the resolution of the New World monkeys. And we actually found that that differed across inference method, not filtering approach. And so what I mean is that when we used concatenated maximum likelihood, it didn't matter whether we used only single copy genes or a branch cutting approach, we inferred the exact same tree. When we used Astral or Astrid with maximum likelihood gene trees as input, we got the same tree. But when we used SVD quartets, we resolved a slightly different tree. And when we used maximum parsimony or Astral and Astrid with maximum parsimony gene trees as input, we found a third topology. And so it appears that this difference is due to something other than the inclusion of paralogs. So both our simulation study and our empirical investigation suggest that species tree inference is accurate in the presence of paralogs. And I didn't show this for the sake of time, but we also did this in several other groups, including plants, fungi, and a larger data set from vertebrates. And we found across the board that including paralogs, especially using those branch cutting methods to extract orthologs from larger gene family trees rather than relying on single copy genes, really did not negatively impact our inferences. And so this is really exciting, I think, because it means in groups where we can't find single copy genes, we can be confident in inferences made from data sets that include paralogs. It also means that we might not have to worry as much about the fact that it is hard to remove paralogs from our data sets, and we might be leaving them in often. Of course, things could be more complex in other empirical systems. And so I mentioned at the beginning that my favorite system is terrestrial gastropods, even though I'm not talking about them today. And in terrestrial slugs, there's a single published genome. And so when I work on these groups, I don't get to work with whole genomes and coding sequences. I have to work with transcriptomes or sequence capture data sets. And these data sets could be a lot messier, and future exploration should focus on how paralogs impact inferences in these types of data sets. It's also true that some groups have undergone more recent whole genome duplications, and that may include terrestrial gastropods. There's a hypothesized whole genome duplication event. And this could complicate inference when we have lots of paralogs. Finally, the last thing I'm going to say about in this phylogenetic section is that I told you, I told you when I started that gene trees disagree with the species tree because of incomplete lineage sorting and introgression and gene duplication and loss. And then I talked a lot about how we could be confident even in the presence of gene duplication and loss. But what we would ideally have would be a method that could consider all of those processes and accurately infer species trees. We don't have that. For computational reasons, it's very difficult to design a likelihood or Bayesian approach that does that and can run in a reasonable amount of time. And so machine learning could be a useful tool for this. Machine learning is helpful when we want to think about really complex models. But remember, in our traditional supervised machine learning framework, the first step is to simulate data under all of the models. But in phylogenetics, the model space is huge. So even with a small number of taxa, we end up with millions of trees that we need to look at and consider. So the model space is too large to use this standard approach for anything more than 4 or maybe 5 taxa. So instead, something we've been thinking about is using approaches that allow for heuristic explorations of model space, like generative adversarial networks or GANs. And so a GAN is a network that actually consists of 2 networks. We have a generator, which makes fake images, and a discriminator, which tries to distinguish real and fake images. At each iteration, the discriminator learns to better distinguish images, but the generator also learns to make more realistic images. And hopefully by the end of training, we have a generator that can make realistic fake images. We took this and modified it by replacing our generator with an evolutionary model. So specifically, we had a tree topology and branch lengths, and then we simulated data and to generate alignments. Our discriminator tried to distinguish our real alignments and our fake alignments. And at each iteration, we used nearest neighbor interchanges and subtree pruning and regrafting steps to propose new topologies. And at the end of training, we inferred that our tree, which was hopefully producing realistic alignments, was the correct tree. Of course, there's a lot of hopefully there, but we actually do find that this works quite well for up to about 10 taxa, though. So for up to 10 taxa, we were able to accurately infer our trees. This is the Robinson fold distance to the true tree. But even with 15 taxa, we weren't doing very well. And this approach was actually quite slow, even when we didn't incorporate things with ILS. When we added ILS, it got a lot slower. And so I don't know if GANs are going to be an answer, but I do think it's important for us to continue thinking about how we can creatively use these approaches to consider some of these complex processes in phylogenetics. And I'm excited about moving forward with other approaches. And so in the future, I want to continue thinking about the data that we use in phylogenetics and how confident we can be in those inferences. So I'd like to look at this in some transcriptomic data sets and data sets from some well-studied organisms like terrestrial snails and slugs. And I also continue to think about the use of machine learning approaches to more directly consider processes like gene duplication and loss. And so today I started by talking about the models that we use in inference and how when those models are misspecified relative to our empirical data, that can mislead our inferences. And so I showed you that when we ignore selection, we can infer migration even when it's not really present. I then talked about some machine learning approaches that I think have some promise for making accurate inference, even when we aren't able to accurately specify our models. And finally, I talked a little bit about the data that we use in phylogenetics and how we actually can use data sets that include paralogs to make accurate inferences. Genomic data, I think, have revolutionized the way that we study diversification in phylogenetics and population genetics. But when we have data from non-model systems or even model systems, inference can be difficult due to model violations and limitations in data quality. But I think by carefully evaluating our existing methods and developing new methods when we can arrive at robust conclusions about the factors that drive diversification in these systems. With that, I will say that my lab has just started at Mississippi State. So if you know someone who is looking for graduate school, I'm recruiting students. And I'd like to thank a lot of this work was done as part of my postdoc work in Matt Hahn's lab at Indiana University and alongside some collaborators at Rice. And I'd also like to thank NSF for the funding for my postdoctoral work. And I should have time for questions. Thanks.
Well, thank you to Megan for a really excellent presentation. I'd like to invite any questions from our Zoom audience. If you have them, please put them in the chat. And also any questions from our in-person audience, although I'll remind any undergraduates attending the private session to please hold on to your questions for that private session. So we'll see if there's any in the chat and I'll read them out if they are. If there are not, right now, I'll start off. So Megan, when you're thinking about why paralogs, people have been in general so wary about including paralogs in phylogenetic studies. I think this is mainly a historical thing, like the problems that it caused for theoretical genetics. Have our tools just been relatively insufficient when phylogenetics started as a burgeoning field, and now that we have better tools, they're just less of a problem? Because one of the big questions I come away with is if you're showing that paralogs don't seem to be an issue in so many systems, why is there so much, fervor and concern over including them?
Yeah, that's a great question. I think there are two parts to that. So the first part, let's see. Sorry. Okay. So the reason that people are really concerned about it, right, is because if I had a single gene and it happened to have a paralog in it, and I inferred the tree from this gene, and I would find that A and C were sister, right? And that would be wrong. But I think what we have come to appreciate more is that there's lots of discordance due to other things too, like incomplete lineage sorting. And so a lot of the methods that I'm comparing are robust to incomplete lineage sorting. And this means that these methods acknowledge that there's going to be variation across our gene trees. And it's actually, for some of those methods like Astro, we can show that the same things that make it work when we have incomplete lineage sorting causing heterogeneity mean that it's going to work when we have gene duplication and loss, just because the most common quartet is still going to match the species tree topology. And so some of it's because our methods do incorporate heterogeneity, but I also showed you that concatenated approaches work fairly well. And some of that is because in the past, if I had 10 genes and one of them was a paralog, that might be a problem. If I have 18,000 genes and one of them is a paralog, it's not going to matter nearly as much, right? So the overwhelming signal is often still going to capture the species history. So it comes from the fact that we have more data than before, and we have better methods for accommodating heterogeneity than before.
Yeah, that makes sense. So I have another question. It's about your machine learning section. I'll say up front that I'm not a machine learning person, but I'm curious, when you're comparing those simulated data sets, when you're developing those training data sets, do you envision that tool as an opportunity to identify the types of features in sequence data that could cause the problems of selection that, theoretically we know exist, but do you envision that approach as a tool to actually quantify the influence of, different strengths of selection in different evolutionary histories and so on?
Yeah, I mean, I think that's a good question. I don't know. What I do know is that we can use those features that we're extracting to try and ask for any certain data set, like how poorly does this data match our prior, right? So that might allow us to learn something about the processes in that system. There's a lot of work being done to make machine learning algorithms less of a black box, so that perhaps we could understand which features of our sequence are actually driving these differences between our source data, our training data sets, and our empirical data sets. So I think there's a lot of promise there that hasn't been realized for sure.
And do you feel maybe that you're kind of on the hunt to look for lineages where paralogs do cause a problem for these inferences, and maybe that there's some interesting biology behind paralogs that we could, or that you could uncover using these approaches?
Yeah, absolutely. I think it's great to try and find the systems that are the exception to the rule, right? Because we might learn something about that system. We might learn about some like weird process that's happening in the retention of gene duplicates in that system or something cool. Or we might also learn when not to trust our results so much, right?
And so we do have a question in the chat, which I will read out from Elliot Sober. Early in your excellent talk, used a likelihood ratio test rather than AIC or BIC. what motivates that choice?
Great question. Since we're just comparing two models and one is just, I mean, the divergence only model is just a model where we've constrained our migration rates to be 0, a likelihood ratio test is appropriate. If we wanted to compare more models or models that weren't nested, then we could use AIC or BIC because these methods are allowing us to calculate the likelihood. So it would be straightforward to use that approach to compare more models or not nested models, for sure.
All right, so I'm sure our undergraduates are chomping at the bit to ask you all kinds of questions. So if we have no more questions popping up in the chat, I'd like to ask you all to join me once again in thanking Megan L. Smith for a wonderful presentation.
Thanks for having me.
Again, we'll see you in a minute. We'll be in another room. So let's see what time is it? Let's reconvene around 1:30. Give you a brief break too.